So welcome everyone back after lunch. So our next speaker is Jacob Korn, who is currently professor of genome biology at the Swiss Federal Institute of Technology in Zurich. Originally, Jacob got his PhD from the University of California at Berkeley in 2007. After that, he moved to the University of Washington in Seattle to do a PhD in computational protein design with David Baker, where coincidentally him and I overlapped for 2 years because I was doing my PhD there, he was doing his postdoc there. So we ended up in the same field again, coincidentally. And after the postdoc, Jacob went to work as a group leader for Genentech first, in San Francisco from 2010 to 2014. And so was at least geographically close to the CRISPR Big Bang, which happened there in 2012. And consequently, he then actually joined the Institute of Innovative Genomics Institute at the University of Berkeley. That was founded shortly after as a scientific director from 2014 to 2018, and then moved to Zurich. So Jacob is actually a good example of a scientist moving back and forth between academia and industry. So his research since 2014 focuses on molecular mechanism of Cas nucleases, as well as their interplay with DNA repair pathways, and also looking into approaches of how to boost rates of HDR. So welcome, Jacob.
So thanks very much for the kind invitation to talk to you guys today. I'm going to talk about genome editing from a maybe different lens than you're used to, because I'm going to be talking about how do we make genome editing better by listening to the cells. I think that massive advances in all sorts of technologies, whether they be different types of Cas enzymes, nucleases, base editors, prime editors, have driven the field forward together with advances in delivery technologies. But the thing that has really lagged behind is the understanding of what's happening inside cells and bodies when we put these things in. All that these guys do is make damage. They cause damage to genomes. And then it's up to the cells and the organisms that host those genomes to take care of it. And very often when we think about this process, you might be surprised to learn that when people talk about, for example, trying to edit the brain for Huntington's disease or edit the liver for hypercholesterolemia or even the bone marrow for Fanconi anemia, very often everybody starts with the exact same tool. They just treat every single tissue and every single cell exactly the same. And they say, well, hopefully it works. And part of what we try to do in my lab is try to take a little bit more of a biology-based approach and try to understand how different cells and different disorders might react differently to different type of genome editing reagents. And in part, we try to do this by understanding what's going on inside the cells. So I think most people have seen a figure like this by now, in which you have a Cas nuclease. It creates a double-stranded DNA break, and that can be corrected in a couple of different ways by the cell. You could have a single-stranded or double-stranded DNA donor, so through a process called homology-directed repair, the cell can copy off that and precisely repair the break. On the other hand, you have non-homologous end joining or alternative end joining, and that can lead to short insertions and deletions. And that's error-prone, and it causes disruption of a sequence. So in human cells, NHEJ or alternative end joining is extremely active. It doesn't have to be this way. If only we were yeast. So the yeast folks, when they do Cas research, they actually do HDR. They even do HDR to do knockouts, because HDR is so effective in yeast, which is a eukaryotic cell where most of our knowledge of DNA repair is based on, that when they want to do knockouts, they do HDR with stop codons. Okay? The situation is different in human cells. Okay? Instead, in human cells, HDR is very, very, very inactive. And in both humans and yeast, it's quite cell cycle limited. And so why is that a problem? There have been a lot of developments in terms of other editing technologies, but HDR remains the most flexible approach to genetic sequence alteration. And prime editing and base editing are ways to try to get around the fact that HDR doesn't work quite as well. So with HDR templates, you can make point mutations, you can make precise deletions, you can insert small cargos anywhere you want, you can insert large cargos. The sky's the limit, really. So you can do all of this with a single nuclease. But the problem is this process is very, very ineffective. And so kicking things off, a postdoc in the lab tried to ask why this might be in primary cells. And she was working in hematopoietic stem cells, building on something that was quite curious that we saw. So just to give you a background on hematopoietic stem cells, they actually toggle back and forth between, think of it as two modes of cell cycle. on the one hand, they're mostly quiescent. Most of the cells in your body are quiescent. They're not cycling, they're just hanging out doing their job. And for HSCs, that's involved in maintenance of the stem cell state. On the other hand, HSCs also need to be able to divide, to renew. Now, through a lot of fundamental work in a variety of organisms, we know that homology-directed repair happens right around in this SG2 phase. And so Jenny asked the obvious question, could it be that the problem with editing in HSCs has to do with this balance, where cells are mostly outside of the state where they need to be for HDR, but if we put them into this state, they wouldn't then be able to maintain themselves. So one of the first things that got us interested in this problem was something that we saw when we started editing so-called CD34 cells. This is sort of a bulk enrichment that you do from bone marrow. And if you take CD34 cells and then you edit them with an HDR donor, and then you break them up into their constituents using flow cytometry into, say, progenitors, slightly less differentiated MPPs, and the most primitive HSCs, you see that different cell subsets have higher levels of HDR. So on the y-axis here is the ratio between HDR and NHEJ. So within this one parent population, different cell subsets do more or less HDR. And if you take just this HSC subset, so these guys are all immunophenotypically HSCs on the surface, and then you further break them up based on cell cycle, you can see that the only immunophenotypic HSCs that did HDR are the ones that are not in G0. So all the cells that are in G0, which are the ones that engraft, and the ones that are going to maintain themselves in the graft, didn't do HDR. And so this is the fundamental problem. We can do HDR, but we can only seem to do it in the cells that are cycling and are not going to be contributing to the final graft. So Jenny turned to it's a GSK mTOR and sort of media cocktail, which we called XRC, which was originally developed to keep cells in quiescence. So this was originally developed to put HSCs into and keep them quiescent for a long time. And Jenny decided to use it for a different purpose. Instead, she would sort of boost the cells with a little bit of a cycling cocktail and then put them into a quiescence media. And when you do that, you can see that cells, HSCs, start quiescent. If you just let them go for a while in culture, they all start cycling. And then if you put them back in a quiescence cocktail, they requiesce. Now, we want to make sure that they're still HSCs, and so Jenny did a lot of single-cell RNA-seq, showing that the HSC mixture starts off as HSCs. If you just let it go, it differentiates into all kinds of different cell types. And then if you put it back in this quiescence cocktail, it goes back to being, it doesn't differentiate, it stays HSCs. And so the question now is, if we do HDR by sort of boosting those cells, letting them go through a permissive phase of the cell cycle, and then putting them back into quiescence, does that affect things? What we see before transplant is that everything looks the same, okay? And that's because these are short time points. So you edit the cells and you count alleles, and you say that everything seems to be doing a certain amount of HDR, okay? Then you take these cells, you put them into mice for 4 months, and then you ask which cells have HDR on the other side. And you can see the HDR levels drop dramatically if you just edit the cells and put them straight into mice. But on the other hand, if you take those same cells, they've cycled once and now you requiesce them so they can contribute to the graft, HDR levels are very high. I want to point out these cells are all treated with the exact same nuclease, the exact same electroporation, the exact same conditions. Everything is exactly the same. The only thing that is different is how you treat the cells, okay? And so I think that that's sort of the first vignette that I want to share to say cells matter. I think this is a lesson that we all kind of know in the back of our heads, but it's very easy to forget this in the rush of genome editing. Nucleases, editing technologies, it's all very cool. I need to pick the right one. Your cells may matter actually even more than any of that. You can, of course, play with different types of editing technologies. There are a variety of ways that people have come up with to reduce NHEJ, fuse different things to Cas9. These are all compatible with this. And that's actually one of the benefits of knowing your cells well, is that you can then use different technologies as long as you know what your cells are doing. So we're able to get a certain amount of HDR, and we're able to enrich it now in the real primitive HSCs, which have always been hard. But the question is, can we actually boost basal HDR? Can we boost HDR itself? And for that, 2 postdocs, Bika Wienert and Chris Richardson, got started by doing a screen. The system that they did was they did CRISPR-I first. In CRISPR-I, you're knocking down genes, not making cuts. And then they made a library of cells that knock down 2,000 different genes in a pool. All of those cells express blue fluorescent protein. Into those cells, they introduce a Cas9 RNP. So this is a separate Cas that you introduce by electroporation. So one gene is being knocked down by a CRISPR-I construct, and then a different gene is being cut and knocked out. Okay? In this case, we're targeting the BFP with a donor that turns the BFP into GFP. And so now it's a pretty simple flow screen in which any cells that are unedited are blue, they're BFP. Any cells that got indels are dark, we cut right on the chromophore. And any cells that got HDR turn bright green. And you can use this, knocking down any one of these 2,000 genes. We just took every Go term related to DNA metabolism here, and look for things that either promote HDR or repress HDR. I want to point out this is about 1/10 of the human genome. We've now repeated these screens genome-wide, and we've found some very interesting things that regulate DNA repair that we've tracked down that are not annotated as DNA repair genes, but in fact play major roles in DNA repair and genome editing. So what did Bika and Chris find from this? Well, first off, there's a variety of things that if you knock them down, you get much less HDR. And so we found things that people normally think of, PALB2, BRCA, HDR complexes. We found the Fanconi anemia complex, and this led to some of really great collaborations with Paula, in which we learned, we sort of started thinking about the Fanconi anemia complex because it plays such a huge role in human genome editing. But on the other side, these are genes that if you knock them down, they increase HDR rates. And so in orange, when I'm pointing out are genes for which there are known small molecules. And so we started looking at these because we wouldn't, from a therapeutic or maybe even preclinical point of view, we don't want to have to make one genetic model to test a different genetic model. We wanted something that we might be able to introduce in a small molecule fashion, and ideally is something that would even have been in the clinic before. So what I'm going to tell you about much more is our work with CDC7. CDC7 is a kinase that phosphorylates the MCM complex that's involved in replication elongation. And so continued phosphorylation of MCM is required to keep driving cells through S. And so if we use this compound, XL413, which has been in trials to inhibit CDC7, we get a very different cell cycle phenotype than people had previously been looking at with so-called S-phase inhibitors. With things like acetylcholine, hydroxyurea, you get these classic S-phase blocks. This is DNA content versus count flow cytometry. So things that are dividing are over here on the right-hand side. So you block with acetylcholine and hydroxyurea. On the other hand, XL413 looks very different than the other ones we've been looking at, in that cells pile up in S-phase. And this got us excited because S-phase is where we want to be. Instead of blocking before S-phase, we're now slowing down S-phase and spending much more time in S-phase. And so we did a timing experiment because the cells matter. When it comes to small molecules, you don't just throw things on your cells. You think about it and you say, what am I trying to do? What's the desired effect biologically? And what we did was we said, well, what's the level of HDR, as in this case measured by a GFP reporter, if we just throw the compound on cells? And then what if we do it in a timed way? What if we first do the editing and then add XL413 to make all the cells be in S while they are repairing the break? That boosts HDR quite substantially. If, on the other hand, you give the exact same compound, but you pre-treat the cells with that compound, and then you edit and you release, now HDR goes down. Okay? So again, it's not about just running small molecule screens and trying to blow through as many compounds as you can. Biology matters, and you have to think about what is the effect this thing is happening. How do I get the cells into exactly the right state that we want in order to boost HDR? So how well does XL413 work? XL413 increases, provided it's given on the right timing, and increases HDR somewhere between 2- and 3-fold in a variety of loci. These are 3 different loci that I'm showing. We've tested a lot more. A variety of immortalized cells, primary T cells, iPSCs, CD34 HSPCs, and it does so for SNPs, epitope tags, and even introduction of larger cargos like CARs. XL413 is also nice because it increases homozygous editing. And so If you look at ploidy of edits, or sorry, not ploidy, but zygosity of edits, here without XL413, you get mostly heterozygous edits and some unedited. And then here you've got a little bit of homozygous, mostly heterozygous. And if you add XL413, it boosts the overall number of alleles, and that translates into being able to isolate homozygous edits much more effectively. So we're pretty excited about the use of XL413, but the thing that remains is that even if you're using XL413, all these blue bars here are heterozygous edits. So there are cases where one allele is HDR and the other allele is not. And if you sequence these, the other allele is actually indel alleles. So Cas9 is so active that what you get is not one HDR and one unedited. Usually what you see when you do these editing experiments is one HDR and one indel. And that's a problem because the indel allele, it can be difficult to do something with that. You may be haploinsufficient. And so we asked, what could you do with that indel allele? One thought that we had was, could we maybe retarget that indel allele? So the idea is, if you make insertions and deletions that come out of here, these sequences are not necessarily random. They do have a defined sequence. And if you sequence the cells, what if we instead targeted these with new guide RNAs, went back to the beginning, and now we're going to get an additional chance to do more HDR. So this work is driven by postdoc Eric Aerd, a really fantastic master's student, and a bioinformatician, Marcus Schroeder. And so we call this recursive editing because you actually get to do this multiple times if you want to. You can do it through multiple levels and just keep driving the cycle. Every time you drive the cycle, you go to the same product on the HDR side, and you're just retargeting the indel alleles. So we started off with a locus that we had an idea would work. The reason we went for this locus is that it has a single indel that accounts for about 50% of the indel alleles in the so-called first round. So in terms of terminology, we call the starting wild-type allele level A, first round of indels B with alleles 1, 2, 3, 4, and then C would be anything downstream. Okay? So we asked, what happens if we start targeting these alleles with new guide RNAs? And the answer is, here in this plot, you can see that HDR goes up quite dramatically. So this is the ratio of HDR to NHEJ, or HDR to indels, and on this side is just pure HDR. Okay? So we start off with one guide RNA getting 15 to 20% HDR, and HDR is in the minority relative to indels. And as we start adding in new retargeting guide RNAs, the absolute HDR level can go up to 60%, and you're getting twofold more HDR alleles over indel alleles. You're converting directly indel alleles into HDR alleles. And if you throw in all of these bottom level guide RNAs, but don't throw in the top level guide RNA, you get zero editing whatsoever, showing that you need to initiate the cascade in order to get any effect downstream. Okay? Looking at the sequences, this is what happens with just the initiating guide RNA. This is that very prevalent one base pair insertion, this one right here. And so we target with this first B1 guide RNA, and that gets directly converted. So if you throw in a B1 targeting guide RNA, this red part of the pie goes to almost nothing, and it converts almost all of that into HDR with a couple of other alleles. We can do that with B2 as well. Now the A1 doesn't go away, but instead the B2 goes away. Okay, so we have molecular control over each of the indel alleles. And this works at this locus using the guide RNAs that we had tested in a variety of cell types. So we're just targeting new alleles. It doesn't seem to matter in this case what the cell type is. It matters that we know what the cell did with that. Now, of course, we're doing a lot of cuts here. And so we wanted to know that this didn't lead to adverse events. So if you take either a promiscuous DNA damaging agent or a different Cas that cuts at multiple places throughout the genome, you can see activation of p53 as measured by p21 expression. And so a toposide is through the roof in terms of amount of p53 activation. A very promiscuous RNP gives you quite a bit of activation. But if you are using a single guide RNA together with these, 2 or so extra guide RNAs, you don't see much of an increased activation of p53. And this kind of makes sense. If you are hitting with something that cuts 20 times, yes, your cells know about it. But it may surprise you to know that you guys are actually undergoing double-strand breaks constantly throughout your genomes, constantly. In fact, every time that any one of your cells replicates, it has a chance of going through something called replication fork collapse, which it looks like a double-strand break and is repaired in the same way. So cells are built to deal with double-strand breaks. They're not poison, and having a couple extra is not really that much of a bad thing. Having too many is a bad thing. I don't want to give the wrong impression. Okay. We can also look at large deletions. So we used PacBio sequencing to look at very long reads and ask, are these multiple cuts giving us lots of larger deletions? Hard to see here because there's not a lot of large deletions, but if you zoom way in, you can see that, yes, we can see large deletions with a single guide RNA, but you don't get more large deletions with multiple guide RNAs. So the message is, it is true, Cas can cause large deletions. We see the levels of this with a single guide RNA vary between 0.5% and 10%. It depends a lot on the locus and the context, but doing multiple cuts doesn't do anything. Doesn't do more. So, great, it seems to work. This is where things were stuck for a long time because it worked, but it felt very dissatisfying because you needed to know exactly what the allele that came out the other side was so you could retarget it, which meant that you basically had to do one round of genome editing, sequence, then come back with new guide RNAs, and it was all very cumbersome and really painful. Enter this fantastic master's student, Lucas Muller. He wrote a piece of software called Retarget, which is an extremely efficient and extremely smart way of doing a very fast search to predict guide RNAs using tools that had recently been developed by Leopold Partz and David Liu. So what do? We start with the genomic region of interest. This can be a genome or it can be any arbitrary sequence. You generate guide RNAs by doing just an arbitrary PAM search. You score them using a variety of metrics that can be plugged in to this algorithm for predicted efficiency and predicted off targets. You rank order based on which ones seem most promising. You then, for each one of those, predict what the indel outcome is going to be with predicted frequencies associated with them. And there's a variety of algorithms. We like Lindell and Delphi to predict those. Okay? You then filter those based on indel frequency. You don't want to go for sites in which the indels are evenly distributed. You don't want a bunch of like 2% this, 2% this, 3% this, 3% this. You want sites where one predominant indel really dominates. You rank everything, you combine this all in a smart search algorithm, and then you find out if you're done. And if you're not done, you go back and you pretend that each one of these indel outcomes is a brand new genotype. And you regenerate the whole process. So now you start to see why this was hard, because you're doing this process in a recursive fashion. That's why we call this recursive editing. And so the search space is very, very big, and it's actually quite a bit of work to get this thing to search smartly. You can put into this algorithm all sorts of things. What is the donor I want to put in? What's the initial guide RNA that I want to start with if you have your favorite guide RNA? I would encourage you to try this out. There's actually a website, recursiveediting.com. This software is fast enough now that you can do this live. So you go to the website, enter your sequence of interest, and it will do all the search for you. So now let's look at sites that we don't know work. Okay? PLCB4 is one site that came out predicted from the retarget software. And so what we had predicted is, here's the wild type. We predict there's going to be a B1 outcome that's about 70%. We're going to target that, and then we have another site that is also stereotyped. And indeed, if we do just one guide RNA alone, we get less than 10% HDR. And by starting to add in recursive guide RNAs, we can boost up to almost 50% HDR. And remember, we are directly converting indel alleles into HDR. So indels are going down at the same time, and we're converting them into HDR. So not only is this sort of a nice to have sort of thing, but we're turning deleterious products into things that we want. It works at another site. It doesn't just work for small things like SNPs and those small deletions. Here we can put in an entire GFP, in this case at a stop codon, and we can do this at a variety of different loci, histones, FBL, and RAB11A, and we get nice increases in HDR at all of them. We can also insert track we can insert a CAR into track in T cells. And so this search is fast enough and this works well enough that at this point we said, well, maybe we can make people's lives a little bit easier. Maybe they don't have to do recursive editing using retarget on that website. And so we actually pre-computed several databases. First off, we pre-computed tagging reagents for every single ORF that we could in the human genome. So if you're interested in tagging any gene of interest, I would encourage you to check out this database. We are able to generate potential recursive editing reagents for about 80% of all ORFs, both start and stop. We went through all of ClinVar, and we were able to generate potential recursive editing reagents for about 85% of all predicted pathogenic ClinVar mutants. And then we went genome-wide, and we said, what if we treat this like a safe harbor? What are the absolute best recursive sites in the genome? And we sort of culled that down to a top decile. And so again, if you want to try this on your own sequence of interest, I keep hitting the wrong button. If you want to try this on your own sequence of interest, please head to the website, but also feel free to check out the databases. This was published just about a month ago, so please check it out. Problem comes, what if we got really good in increasing HDR, but there's a problem in the cells itself? So what if the very cells that we're in can't do HDR at all? Not because of cell cycle or anything like that, but because of the very reason why we would want to edit them. So as you heard from Paula, Fanconi anemia cells are deficient in HDR. And in fact, the reason we got involved in FA in the first place is this result that we had in 2018, where we went searching for things that impacted CRISPR-Cas HDR, and all the top hits were Fanconi anemia genes. And so it turns out that if you have Fanconi anemia, any one of these mutations, it's very, very, very hard to fix that problem with CRISPR-Cas HDR. Paula told you all about the indel-based approach. This is quite effective. The therapeutic edits do expand, but it works in a limited number of genotypes. Now, different editing technologies leverage different pathways. Again, I sort of want to say that again, because the way editing technologies often get presented is this sort of magic pencil, and we editors are just as liable for spreading this rumor, this sort of fallacy. HDR depends on multiple things in the cell, right? It's not just direct editing. Same thing with base editing and prime editing. So base editing is probably related to base excision repair and mismatch repair. And I know Alexis Comore has a story that she's been working on about the cellular determinants of base editing. Prime editing, we know, is related to base excision repair as well as mismatch repair. And David Liu had this very nice paper, as did Joanna Louisou, about different factors that are present in all cells that if you knock them down will increase prime editing. And so, in collaboration with Paula, Erman, Sebastian, and Alexandra asked, maybe we can use one of these alternative ways to edit FA cells since they can't do HDR. So we started off in a FANK-A genotype using a lymphoblast cell line that we got from Paula. So this guy is deficient in FANK-A. That leads to a stop codon right here. Now, we can't directly reverse the stop codon just because of the type of mutation that this is. But what we found is that we could go from glutamine to stop to tryptophan. Turns out this position is quite unconserved, and there's actually 2 species of otter that have a tryptophan at that position. So I know we're not otters, but stay tuned, okay? So we said maybe this guy would be promiscuous enough that if we base edited to a tryptophan, it wouldn't be all that bad. Our first hint that that might be okay was when we started base editing, and we got low levels of base editing with plasmids around 5 days. This is adenine base editing. And then over time, the cells expanded. You heard from Paula that wild-type cells have a very large proliferative advantage over FA cells. And so we thought, well, presumably this tryptophan is not total poison. It's at least better than a stop codon because these cells are able to expand. I put this slide in because the question was asked earlier, what is the editing window? With these particular editing reagents, this is sort of a classic way of looking at editing windows, first shown by David Liu when he started describing base editors. So here, things that get changed would be in red, and you can see that there's very little outside the window or very little unintended changes at this particular locus, and none of them really seem to expand into anything. Now, if we do the base editing and then let the cells go for 30 days, we can see that they have recovered MMC resistance. This is just titrating in amounts of MMC. The base edited pool is all the way over here with wild type. They also re-express FANK-A, and if you expose them to MMC, they've regained the ability to ubiquitinate FANK-D2. This type of base editing strategy also works, but at low efficiency in a couple of other genotypes for FANK-A and for FANK-T2. So base editors can be used, and they can be used to edit FA alleles. But starting at 5% and waiting a long time to expand is not ideally where we'd want to be. So we asked, could we get better? And so we iterated a lot of things. How do you, trying different base editors, trying location of the guide RNA, trying the format of the Cas, the format of the guide RNA, delivery conditions, etc. And where we eventually ended up is here. So we have now mRNA for either ABAMAX or 8E, ABA8E, which is a very hyperactive variant developed by Jennifer Doudna and David Liu. And now instead of showing you day 30, I'm showing you day 5. So this is equivalent to what we had down here. Now we're getting almost 80% base editing at day 5 using ABA8E. If we compare ABA8E and ABAMAX, ABA8E is much more active. However, it does not seem to come with increased off targets, at least at this particular site. So for this particular guide RNA, ABA8E doesn't necessarily give you a lot of dirt. But if you choose a dirty guide RNA, both ABAMAX and ABA8E will give you some off targets. And whether or not those off targets are a problem kind of remains to be seen. It depends on where they would be. In this case, we don't think they necessarily are going to be a problem, but this allele is very cleanly converted. We've tested many more than these 8 off targets now for the top one. I think we're up to 30 or 40. For both ABAMAX and ABA8E, you start very MMC sensitive, both for a FANK-A allele and for a different FANK-A allele, FA55, and then a different mutation. And then editing increases MMC resistance. Very importantly, the data that I showed you previously was editing for 5 days, letting the edited cells grow out so that the guys with the fitness advantage take over the population. This now is showing you the MMC resistance in just 5 days after editing, so almost immediately after doing the base editing. And so at very, very short time points, you can get phenotypic correction. So it looks like genotypically, I switched this around. This should be greater than sign the other way. So 8E gives you much more genotypic correction than ABAMAX, but the phenotypic restoration seems to be about the same. And then finally, does this work in HSCs? So indeed, base editing, as you saw, can work in healthy donor HSCs. That was data that Paula showed in her talk. This is now showing CD34 FA patient HSCs. 5 days after editing, and then you genotype, you can see very high levels of A to G conversion in these HSCs. CD34 cells, you can get around 60% or more editing. And then in longer-term colony-forming units, almost all the colony-forming units are edited. And again, that's because this takes longer to make the colony-forming units, and the edited cells have a huge advantage relative to the patient cells. And then finally, the real cherry on the top of the sundae from Paula's lab was taking these cells and looking at MMC resistance, and you can see that phenotypically FA cells are really killed by MMC. So survival is almost 0, but in a variety of patient settings, 8E strongly increases MMC resistance in these primary cells from FA patients. So pretty excited about that part, and I'm excited to keep working with Paula on moving this forward. So what have I told you about so far? HDR is extremely flexible, but as I think anyone who's tried it knows, it can be very inefficient. You can increase HDR rates, but you can't do it just magically by trying a bunch of different tools. You have to do it by listening to the cells and trying to figure out why is it that HDR is low in those cells. I told you about 3 stories about controlled cycling and quiescence, slowing of S phase to increase the proportion of cells that are in the permissive phase of the cell cycle, and then also recursive editing to directly convert indel alleles to HDR. And then finally, I told you about saying, well, what if we can't do HDR at all, and optimizing base editing to be effective in very sensitive HDR-deficient HSCs. So I hope that I've convinced you that cellular context determines everything. Genome editing is not a magic bullet in which you just, or a magic pen where you just go in and directly make the edit that you want. It all depends on the cells. We need to know what the cells are doing. Genetic disorders often impact the very pathways that make these edits. And one of the things we're very excited about in the future is looking much more deeply into the cellular context question, looking at how different cells in your body can repair different types of genome editing. And just to give you sort of a teaser for that, why this would be important, if you think about the repair propensities of your body, think about your skin, which is exposed to all kinds of UV, and so expresses all kinds of genome editing reagents that are related to that type of DNA damage. Compare that to your liver, which is exposed to all kinds of toxins like aldehydes, and so expresses huge amounts of repair enzymes to take care of that kind of damage. You are not yeast, you are not a unicellular organism, and you are exposed to different types of stresses and different types of toxins all the time. And I think that knowing how different parts of your body and different cell types in your body are specialized to take care of that damage will dramatically push forward our ability to make exactly the type of edit we want at exactly the time we want. So with that, I just want to finish up. I hope I thanked everybody who was really involved in the work from the beginning. These guys are all the real heroes of these stories. Thanks to incredible collaborators, especially Paula, for these really precious patient cells and for working with the patients, and fingers crossed on moving some of the stuff forward. Thanks to generous funders, and thank you very much for your attention.